Hi, everyone. Welcome back to part two of our question and answer session as a follow-up to our panel discussion, BRD, if everyone vaccinates, why is it still a problem? As mentioned, we had a really great response to the in-person session as well as the live stream session. And so there were a lot of questions that were posed to the panelists that we didn't get a chance to get to, and so they're working their way through these. It's been a great discussion so far. Our three panelists, Dr. John Ellis from the Western College of Veterinary Medicine at the University of Saskatchewan, Dr. John Richeson, who from West Texas A&M University, and Dr. Amelia Willems from the College of Veterinary Medicine at Mississippi State University. And my name is Paul Morley. I'm a professor at the Vero program at Texas A&M University, and I'll be moderating our discussion. So, going right back into it, the next question goes to John Ellis. John, the question says, we haven't seen a new or updated modified live vaccine in quite a while. Is that because we've maxed out the vaccine potential or we don't see much viral change in these products?
Well, I guess the first thing to acknowledge is that we are, a neomaniacal species. If it's new, it's got to be better. So I really don't think that new vaccines are going to contribute much to, the situation because I think that the immune failure, for lack of a better term, that we're seeing is more about the host than any kind of antigenic mismatch and inadequate vaccines. And so I think we've got a lot of really good vaccines, and kind of low tech vaccines. I mean, one of the things you could see coming maybe was a self-amplifying mRNA vaccine, say for the bovine influenza virus, and whether or not what happens with that might be a situation where that sort of a thing could be applied again because of the rapid change in the glycoproteins of the influenza viruses that might have a potential. But I don't think that new vaccines are really going to contribute to that much to the situation. We've got certainly, I think, vaccines for the most important pathogens. Well, with regard to the viral pathogens, are good ones. Of bacteria, maybe more chance for improved vaccines there. But for the viruses, I think we've got the most important, as far as we know, antigens and simple modified live vaccines. I think do a pretty good job if they're administered properly.
You bring up a good topic that we really didn't touch on. I was a little bit surprised we didn't touch on it during the panel. Dr. Willems, you want to talk? You want to touch on the mRNA treatments in
Well, thanks for asking, Paul, since that is something we've been working on with some important collaborators. But before we do that, could I just ask Dr. Ellis? I think, the questioner was probably wondering about viruses changing over time. And you, I think, kind of acknowledged in your answer that that's not really a problem for the common viruses that can contribute to BRD. And I know some of the work that you've done with one of the BRSV isolates you've used, some of the work suggested that BRSV didn't really modify over time, if I remember that right. But what do you think about coronavirus? I wanted to ask you if you thought of all the kind of viruses that we use vaccines for commonly these days. I sort of wonder if coronavirus is one that changes enough that maybe we could use some updating, but then people even debate how much that contributes to BRD. So I guess what I wanted to say is, what do you think specifically about viral sort of modification over time, mutations of viruses over time, so they escape the vaccines for BRSV and for coronavirus?
Well, for again, I'll just focus on BRSV because I think that's an easy one. And I had that question from a producer or a veterinarian just last week. And I think, again, the chances of that being a real problem are pretty low because if you look at the F protein, contains the major neutralizing epitopes, there's like a 90, greater than 95% sequence identity amongst the known strains. And so, yeah, the G protein changes some more, but in terms of the main protein that's important for inducing protective response is the F protein. It just doesn't seem to change that much for some reason, maybe somewhat counterintuitively because it's an RNA vaccine. So I think for RSV, probably not that much of a potential for importance of escape mutation. With corona, certainly there's more of a, as you rightly say, there's way more probably chance for that to be occurring. Certainly that was the situation with the SARS vaccines. But the other part of that without getting too much into the weeds is, if you select one protein to target, which is the reductionist approach to vaccination, does that increase immunological pressure? And so are you better off with a lower tech vaccine, which is kind of a Luddite perspective these days? But if you have more of the virus to respond to, it's difficult for the virus to mutate around that, at least theoretically. So, but the short answer to your question is, yeah, I mean, it's a really good question with regard to bovine coronavirus, and part of the challenge model for assessing that.
Yeah, I have to admit, I am sort of suspicious that coronavirus is contributing to an important proportion of the BRD, based on basically some of the published research. But I think it is a little bit controversial. I mean, Dr. Richeson, what do you think about that?
I get that question all the time from producers about, and I guess I'll use BVD as an example, BVD virus, but like the Singer strain, whatever, pick your strain, those were isolates from decades ago. Or the old strains, are they really providing, should we update the vaccines? And I guess my common sense answer would be that, if you were going to get a lot better protection trying to incorporate and make a new vaccine out of a newer isolate, I think there would be a lot of impetus for the companies to do that. There was a type 1B BVD vaccine for a while that was on the market, and I don't remember why, but it's not on the market anymore. But you get good cross protection between 1A and 2A, and with the BVD vaccines, there was an effort to try to have a new isolate and have a 1B vaccine, and it didn't even last on the market. So, but definitely I hear that question a lot from producers.
Yeah, what's missing in a lot of these studies is because driven by the industry is direct comparative studies. And so again, the marketing claim is we've got this new vaccine, it's better. But a lot of times in those claims, there's no direct comparison. Is it really better than the old vaccine? Those kinds of questions are often not there, or those kind of questions are often not answered. What the question everyone really wants to know.
I think it's also kind of unusual when people have what they perceive as a failure of a vaccine to work, for them to then collect the sort of the microbiological samples to know if the virus in the vaccine is even circulating in the cattle or played a role in the disease.
Mm-hmm.
Sometimes that happens, but it doesn't happen very often. Like I would think with BVD, if you had cattle that you thought were well vaccinated for BVD and then you were having more BRD than you expected, did you look for BVD in those cattle? Sometimes people do, but they don't always. That would be the first thing you'd want to know is, well, did they have BVD circulating them in spite of being vaccinated, or did it seem to be playing a role in the disease? And, we all get asked about, is it worth it to do some nasal swabs, right? And the idea being to look for the agents that are circulating in the cattle. And, I think that can be helpful, but the samples, you have to kind of pick carefully when to sample, right? And what to sample. Because I think people sometimes sample cattle when they've been given an intranasal vaccine. That can really complicate interpretation error results. And also sampling animals, like if you're doing samples at necropsy, the virus may have been important 2 weeks ago, but it's not there anymore. So you really need to be sampling some of the cattle that are not dying at the beginning of the problem. And that's the thing that's often lacking when you have these problem outbreaks.
Well, that's a great point in terms of the timing, and a way around that, which has become another Luddite approach, is acute and convalescent sera. right? No one who's doing that anymore. All the diagnostic labs it's all molecular. And so it's real easy to miss a virus. I mean, I know from experimental challenges that are pretty natural, you peak 6, 7 days and the virus is gone after that. And so, so that's where serology can be really valuable in terms of an outbreak situation. If people, just collect some sera on the first visit and then 2 weeks later, some more sera from the same animals and test them both at the same time. If there's seroconversion, even if you don't see the virus, that's a pretty good indication that a virus or a bacteria was involved.
Unless they were vaccinated, right? I mean, that's complicated if they were vaccinated at arrival.
Yeah, true. But um, it still is worth, in some instances, a thought. I mean, it isn't even on people's radar anymore. I think I guess my point is that, and given the kind of just run towards the molecular diagnostics, I think we're potentially missing things.
Yeah, I
Amelia,
still think
you done?
the surveillance research would be very helpful, but like has been pointed out, it's got to be very coordinated and thoughtful, right? And sequenced so that it's useful, I think.
It's a great discussion. Amelia, you artfully did not answer my question about what about the future of mRNA vaccines?
Well, so, and I think you asked me about mRNA therapy, and that's what we've been
I said
working on is mRNA
products. I said
therapy.
products.
Yeah, mRNA. And so, if people haven't thought about mRNA therapy, it's the idea of giving a messenger RNA to induce production of a protein that would have a therapeutic effect. And probably what's being looked at the most right now for in terms of respiratory infection are antibodies. You could actually give a messenger RNA into the respiratory tract to induce really within 24 hours production of antibodies that could be neutralizing. And I think that is pretty exciting and an area that warrants investigation. Actually, I know groups who are working on that. Now, back to RNA vaccines. Obviously, they've been very controversial in the wake of the COVID-19 pandemic. I think that the thing to remember about RNA vaccines is that they aren't necessarily going to be more effective than another type of vaccine, but you can generate them faster if you have a bad outbreak with a pathogen that you sort of have information about in terms of, what's the RNA sequence of the proteins that are important for immunity. So I think the reason that people should try to stay open-minded about RNA vaccines is that if you had another pandemic or a sort of panzootic in cattle or any livestock species that you were trying to bring to a close rapidly with a vaccine, a company could make an RNA vaccine faster than some of the other more classical types of vaccines. So I certainly think it's something we should keep kind of on the table in terms of vaccine technology. But I would say that I don't think an RNA vaccine is necessarily going to be better than one of the kind of old school vaccines Dr. Ellis was referring to, a whole virus, modified live vaccine that can be pretty effective when used correctly, I think.
Okay, thanks. That's interesting. I guess I had thought that the there was a stronger consensus that the mRNA vaccines could generally be more effective. So that's interesting that
I
you
should
say.
probably
So,
just to quickly point out that remember when we give an RNA vaccine, we're giving the RNA sequence for a protein. And of course, you could have multiple RNAs in the vaccine. And that RNA has to be taken up by a cell and the protein has to be made. So it will be made by the cell and either secreted or expressed on the cell surface, kind of like the virus protein would be made, or if it was some other type of agent that replicated inside of a cell. So if we compare an RNA vaccine to, let's say, a subunit protein vaccine, like let's say in the example of BRSV, where the F protein that Dr. Ellis mentioned is really important for immunity, we could either genetically express F protein in vitro and just give the protein, or we could give an RNA for expressing the F protein. And because the RNA would be taken up and expressed by cells, you might have better immunity when you give an RNA vaccine. Having said that, in humans there are F protein subunit vaccines that are having some efficacy, but I don't know if those have been compared side by side. So there are some reasons that RNA vaccine compared to a subunit protein vaccine might be better because of the way the protein would be expressed to the immune response. But when we've got whole live virus vaccines, which are kind of rare in human medicine, right? There aren't very many whole live virus vaccines. We have a lot of those in cattle. There, you're going to get infection and get production of the proteins. And I don't want to get in the weeds here too much. I can imagine circumstances where an RNA vaccine might do better than a whole virus modified live vaccine. And I can imagine situations where it wouldn't be as good.
Okay, thanks. Thanks for sharing your expertise. You're pretty unique in your knowledge around that as it relates to cattle. So, um, John Richeson, do you see a future in using biomarkers such as cortisol or leukocyte profiles to determine when to vaccinate, as opposed to basing it on specific events such as vaccination upon arrival?
I hope so. I mean, it's a monumental task, I think, to be able to really understand the conditions of the animal and have some sort of test to guide decisions and whether you vaccinate an animal or not against what antigens or whatever. But I mean, we've done quite a bit of work with trying to predict BRD with various biomarkers. And the primary product of that would be to facilitate targeted metaphylaxis. So if I have this group of high risk animals, I know that even though I classify the entire population as high risk, there's definitely differences at the individual animal level and risk outcome. And so could I use some sort of test to help me predict BRD and whether or not an animal needs metaphylaxis or not? I think when you get on the vaccine side, it gets even more complicated. And and that was a difficult challenge to be able to predict BRD risk for targeted metaphylaxis. Ideally, we have some sort of test. If you believe that within a high risk population, there's cattle in there that are going to benefit from on arrival vaccination. There's cattle that it may be detrimental to, and there's cattle that it may be equivocal. If I had a test that could accurately tell me that, that would be the golden goose, right? Everybody would love that. I just think we're a long ways away from having that technology. Whether you're using a complete blood count profile or cortisol or haptoglobin or some sort of biomarker, they're just not specific enough or granular enough to give us that information.
Yeah, our diagnostics around who got sick is hard enough, let alone diagnostics around who's going to get sick, right? So,
Yeah, I mean, saying that, like, I think there's a lot of promise with these behavior monitoring ear tags. I don't know, I'm kind of getting off topic a little bit, but uh, there's been a lot of talk amongst all of us about how difficult it is to diagnose BRD, and that's definitely true. We have false positives, false negatives that we know about in the field. I think these ear tag monitoring technologies hold a lot of promise, and I think if and when those do get widespread adopted, I think they will have a big overall impact on BRD mortality across the industry, I hope.
yeah, the sensor technology, we don't know where that's going to take us, right? Whole new world in some of that sensor technology options. John Ellis, do you think BRD vaccines fall short due to weak immune response or the wrong type of immune response, especially under stress?
Well, I guess some variation of the weak immune response, because again, without sounding like a broken record, it's hard. When you're vaccinating on arrival, you by definition are going to get a weak immune response just in terms of the timing. So I think it's simply a timing issue in most situations. Again, I think the vaccines we have are good if they're administered properly and given enough time to stimulate the body to react.
So just to clarify, or just to ask a follow-up, you're not saying that the vaccines are not effective when they're administered on arrival. It's just that timing related to when the disease challenge or when the exposure challenge is happening is too short.
Well, it's a little of both, right? I mean, we talked about it somewhat facetiously in the panel, just stop vaccinating. I'm not recommending that, but I mean, we have to look at the constraints, and we've all talked a lot about this, in the panel and today is we just have to acknowledge what the constraints are and what we're expecting from vaccination. And I think
So go ahead. Sorry.
we're way, our expectations around vaccines, I think, are way unreal in many situations in terms of looking for a silver bullet or a magic bullet in a situation that the biology simply isn't going to give you that.
Okay.
So
vaccination pre-weaning, vaccination at arrival, do those calves, everything else being equal, do they respond equally well to the vaccine?
I would say no. Again, it's in most instances it's just simply, as John would, I'm sure comment on it, it's complicated in terms of the overall response. But if you just look at timing alone, if you're exposed on a truck.
I don't mean related to disease occurrence. I just mean as far as the immunological response. Are you saying that those two calves, the one that's vaccinated pre-weaning and the one that's vaccinated at arrival, that they respond different successfully immunologically?
Well, see, I don't know that that's ever been looked at in any kind of defined way, right? You could set up an experiment to address that, right? You stress a bunch of calves. Well, you'd have to control for exposure. But if you ask the question, does stress alone, acute stress alone, affect the immune response to vaccination? I would predict no, based on everything we know. If it's chronic stress, it's another thing. But the stress in and of itself isn't necessarily going to be negate against an immune response, a protective immune response, if that's the question. So, but then the timing issue. So there's maybe two issues you're talking about: timing issue, the kinetics of an immune response, or the effect of acute stress on that response. So those are two different questions, which could both be approached experimentally.
Okay.
Others?
Yeah.
We've done some research to try to understand that, not claiming that I know all the answers or the research we've done answers all the questions, but I think everybody hopefully understands the concept of if you have a live replicating vaccine, the host effects can change the antigenicity of that vaccine. In other words, there's a TCID for every MLV vaccine, and the infectious dose but the antigenicity of that vaccine can differ from one animal to another based on complicated physiological and immunological conditions of that animal. And so when we're talking about stressed, immunosuppressed animals, my opinion is that their overall immune response is definitely blunted. They're not going to produce antibodies and all the things that stress does to immunosuppression, reduce immunotaxis, reduce phagocytosis, reduce antigen presentation. That's all at play. But the antigenicity of the modified live vaccine is changed significantly, meaning if the animal's immunosuppressed, that modified live virus, that TCID in the 2 milliliters has a lot more replication that's occurring in the immunosuppressed animal than in the homeostatic animal. Therefore, in all the research we've done, looking at like stress versus unstressed vaccinated cattle with a modified live vaccine, we see and it's antibody titer, right? As a proxy, that's the problem because that's just one piece of the immune response. But the antibody titer is pretty strong in response to one MLV dosed in a stressed animal, whether it's from the natural kind of field studies where we've looked at timing of vaccine, either on arrival or delayed 14 or 28 days, or whether it's using some sort of stress challenge, more controlled. You don't see this huge blunted antibody titer response in the stressed cattle because the antigenicity of the MLV vaccine is much greater in that immunosuppressed host. In other words, that immunosuppressed host, their immune system permits those live attenuated viruses in the 2 milliliter dose. They replicate much more because their immune system is blunted and it doesn't stop or control that replication of the modified live vaccine agents in the vaccine the same as it does in a homeostatic host with a normal functioning immune response. And it's very complicated. It's hard to understand, but because it's the opposite with the killed vaccine that does not replicate. We have clearly seen in those same studies looking at a killed antigen, it's much less and again, it's titers only, but the titer response to a killed vaccine in a stressed host is much lower than a non-stressed cohort.
Okay, very good. Amelia, did I ask the question? Many producers send their animals through the production system with very little clinical disease. What are they doing right? Did I ask that question?
No,
Okay,
we haven't done that.
okay.
So
I'm
it's
asking that
a
question.
so I think that is super interesting, kind of related to what we talked about earlier, that some cattle seem to resist getting sick, even in our imperfect system. And what's happening that's right? Is that because of something the producers did, something about the cattle themselves? Um, so, you know, I I think that we've all agreed that there is some evidence that vaccinating properly before the cattle are shipped can be related or associated with less disease. So I would feel like I need to say, well, some of these sort of success stories are related to being managed properly, most likely. But it's hard to find the data that prove that. The study set up properly to prove that better nutrition pre-weaning is associated with better health in the feedlot. You know, while there are studies showing that preconditioning can be associated with better health, certainly we all have heard of situations where cattle were apparently well vaccinated and didn't have good health. Interestingly, some of the studies that look at preconditioned cattle showed that some of those cattle arrived with no antibodies, right? To the things that were supposed to be in the vaccines they were supposedly got. So sometimes cattle can be supposedly vaccinated, but have no measurable antibodies to what they were supposed to have been vaccinated for. So sometimes people may think they're doing something right, but it's not working. So back to the question. I think that what are these people doing right is a question that merits more investigation. Because I don't like to just repeat the sort of dogma that we've all heard a million times. Uh, I think there's some evidence that proper vaccination, proper nutrition, can be important in sort of supporting health. But I think that we do still sometimes have cattle that don't do well when we thought they would do well, and we have cattle that do well when we think they wouldn't do well, which means we don't understand the whole answer to that question. And then we've always got to keep in mind that we don't always diagnose the disease well. I guess when the cattle die, everybody can pretty much tell that's happened. So if we're talking about outcomes of high mortality versus low mortality, um, we could say, yeah, there must have been something different done about these cattle. But if we're talking about morbidity, I think it's important that we recognize that we don't always identify the cattle that really had BRD very accurately.
Well, and I hate to go back to study design, right? But if we were trying to build evidence on that, if you're trying to build a study around mortality outcomes, it's so much harder because of the low occurrence relative to morbidity. So we have the diagnosis bias in question in there. Yes, they died, and we can classify what they died from, but it's very, very hard to do the study for around mortality because it's a much more rare event, right?
Right. Very good point. I'll just throw out something that I think probably anybody who's listening to this that's experienced knows this already, but in case there's anybody kind of young listening to this, um, some cattle get transferred through a lot of sale barns before they end up where they're going. And I've certainly heard anecdotal reports that make me think that that's an important risk factor for increased disease. This idea that cattle have been transferred through multiple sale barns. I think that some cattle come with quite a bit of pneumonia already present, but we don't always identify that. Personally, I wish that we could really measure accurately how many times the cattle transfer hands before they finally get where they're going. And that was, I think, maybe related to a comment made at the panel that if we had individual ID and we could really track where every animal came from and where it goes, we might get some better ideas about the cause of success and failure.
And there is, you and I are both aware that there is research ongoing that it's initial, it's not tons of research, but looking at systems based questions. So if they go through the auction market or they don't go through the auction market, do we see differences in health outcomes as well as immunological and transcriptomic markers? So there, stay tuned because there is some work on that, and it does seem like that the idea that we're vaccinating at the wrong time, that's can we that's a systems based kind of a question. Can we change the system, or should we change the system, or in addition to whether or not the products work? So yeah. John Richeson, um, if you get most of the cattle in a pen on feed faster, does that drop cortisol and reduce sickness? Or do you get them if you get them straightened out faster? Let's use jargon here, right? Are those cattle less likely to have more disease?
Yeah. I mean, I think the faster cattle acclimate to their new environment, the less likely they are as a population as a whole to get sick. I remember a quote that I think Dan Thompson used to say this all the time, that sick cattle don't eat and cattle that don't eat get sick. And that's where I think, I mean, nutrition, but just acclimation, right? From the ranch of origin into the stocker facility or the feedlot where they're going, the better the group as a whole takes that acclimation and that transition to their new environment, the less chronic stress persists, the faster they get into homeostasis. And feed intake is certainly a proxy for that, right? Because the cattle aren't eating because of all the stress and the inflammation that's occurring from the marketing process, but also a lot of the stuff we give them at processing is very inflammatory, causes a cytokine response. Cytokines certainly impact anorexia and cause animals to lose their appetite. And so, the longer that low feed intake persists, the more likely you're going to have trouble with BRD for lots of different reasons they're very complicated, but certainly true.
So is that
Amelia.
so? If they get on feed faster, okay, is that a cattle issue, or is that a management issue? Is that a system issue?
Yeah, you can mess that up definitely as a manager. I mean, we've looked at some research like with different diets, right? Like these cattle come in and no matter what we do, they're going to have pretty low intake for the first couple weeks until they acclimate and get over their inflammation and adjust and start to get their appetite back. So maybe it's better to give like a higher concentrate, more energy dense, higher protein diet versus a more roughage diet. But the research would suggest that the higher concentrate diet, certainly performance is better, but morbidity and mortality is going to be a little bit higher if you go that route. So, and I've seen that repeat itself across multiple studies. I do believe that's true. So, giving cattle roughage that they're more familiar with, I think there's an acclimation aspect to it, but there's a metabolic aspect to it as well, to where metabolically it's not quite as stressful, I think, versus like asking them to consume their calories from a high concentrate, high starch diet alone.
Okay, that's interesting. Amelia.
Yeah, I really want to ask John to follow up on that because people have said they cattle were put in a position to fail by the way they were managed when they got to the feedlot. Usually, the implication being that they were put on too much concentrate too fast. And I wonder how much John thinks that, how common is it for that to be a primary factor in sort of disease occurrence, do you think?
I think it's a factor. I think I think a lot of it's imprinted before they arrive, but I do think it's a factor. I think if you try to transition cattle too quickly, it can certainly cause problems. It can cause metabolic issues, right, with acidosis, which clinically can look very similar to BRD. I wonder if we're not misdiagnosing BRD and acidosis at times, but it is a factor, but it's also a balance of the system and economics and feed efficiency and, trying to get cattle towards a finishing diet as quickly as possible. Yes, it may impact health a little bit, but there's a trade-off as well. So, but certainly I do believe the work that Lofgren did long ago, that's in the general thumb rule that more roughage is probably means better health at the expense of performance and feed efficiency. So if you do push cattle too quickly and try to transition too fast, I do think you can cause some disease problems.
Great thoughts. Amelia, is the idea that a vaccine can protect an animal from commensal bacteria misguided? Should we be focusing on vaccinating for virulence factors instead?
Yeah, so I think the questioner is thinking about our vaccines for Mannheimia hemolytica, Pasteurella multocida, and Histophilus somni, which can all be found in the respiratory tract of healthy cattle and are generally considered to be part of the normal flora of the upper respiratory tract. So, is it even, are we on the right track trying to develop vaccines against those organisms? Um, I guess I should just say that when people have tested vaccines in challenge studies, that those vaccines can decrease the disease associated with challenge. That's how the vaccines get licensed. But is that really relevant to what's happening in kind of the field? Um, I think that it's a good question. I think that the Mannheimia hemolytica vaccines have been sort of, I think, sort of developed the most different options and studied the most. If you look for clinical trials for vaccines against Mannheimia, Histophagus, I think you'll find that the most that are published, at least, are for Mannheimia hemolytica, and that the leukotoxin, which is a virulence factor, has been shown to be an important component of a protective vaccine. So in the old days, people tried just Bactrians, just kill the Mannheimia and give it, and those weren't very effective. And in fact, sometimes they even made disease worse. And that led to work, some of it, quite a bit of it done at Guelph, but also in other places in the world, in the United States and elsewhere, that led us to determine that the leukotoxin was a really important vaccine antigen. So And in sort of contrast, I don't feel like we've really got that research for Pasteurella multocida and Histophilus somni. You can probably find a few papers in the literature where people have looked at things that Pasteurella multocida or Histophilus somni produce or make could be included in a vaccine that may be effective. But as far as the sort of thorough research looking at virulence factors in P. multocida and H. somni, that are important immunogens, I think really just hasn't been done very thoroughly. And so it may be that we would have better luck with some vaccines against virulence factors for those agents, I think. But I also think that when we vaccinate against Mannheim hemolytica, for which I think we've got sort of the best vaccines for those kinds of organisms, we still see some BRD. It decreases BRD morbidity, but doesn't really decrease it a lot. There have been some meta-analyses on Mannheim hemolytica vaccination, and it has an effect, a positive effect in those meta-analyses where they look at multiple clinical trials, but not a very big effect. And so that may also sort of reflect that it's the host response to Mannheimia getting down in the lungs, maybe being too much inflammation, rather than a pure can the host resist Mannheimia getting down into the lungs that is important.
Thanks.
John or John, either of you guys want to say anything about vaccines for commensal bacteria?
No, I mean, it's somewhat problematic if you think that, I mean, on the one hand, we talked about maybe in a little bit of the panel or before that the disease in Mannheimia is due to the response, right? The neutrophil response. And so that kind of suggests that if you control the Mannheimia load in the lung, then you've got less disease. But then the whole idea is how does it get there and what's it doing? So it's complicated. That's why I like to work with viruses. It's way easier.
Maybe. I will say, Amelia, and you're aware of this because you're part of it. But if we look at Mannheimia, let's just pick on that one. But then also, I can see it with mycoplasma now too. That our group and others are about ready to publish. There's increasing amounts of work that's about to come out and has come out saying that there's greater strain diversity in these bacteria than we ever thought was present, and we can find multiple strains in individual animals. Is that related to a difference in which one cause disease, and which ones are more likely to be found as commensal? There's a little bit of that work, right, that would suggest that the genotype 1, genotype 2 differences in Mannheimia are maybe slightly more common in healthy versus diseased. But additional work needs to be done. But we're about to see more of that. We're about to be dissected in greater detail.
Yeah, for sure. And that will be good to kind of get that information out and then follow up on where that takes us. And you
Yeah.
mentioned Mycoplasma bovis. I specifically didn't mention that as a commensal. In the past, I think a lot of people thought that Mycoplasma bovis was just an opportunist that only caused disease when there was some other underlying insult, like a viral infection. But I know that some of our European colleagues are pretty confidently calling Mycoplasma bovis a primary pathogen these days. And I think that there's a lot we don't know, but I have kind of come around to thinking that, it's interesting. You don't find Mycoplasma bovis on nasal swabs every time you look for them. And if you look for Pasteurella or Mannheimia hemolytica on nasal swabs, nasopharyngeal swabs from cattle, you can certainly find it in healthy cattle. But when we have sampled groups of stocker cattle at arrival, we did a small study a few years ago. I was surprised how little Mycoplasma bovis we found, even in
By
these cattle
culture.
that no, well, we did PCR and culture both. And
Okay.
this was a small study we published a few years ago that when I was at Georgia, we went to, I think, 9 or 10 different stocker operations and sampled 50 cattle. So after the cattle had been commingled and transported, um, and we know there's a lot of evidence that Mycoplasma genus, just Mycoplasma, not bovis specifically, but Mycoplasma the genus, can be found in higher sort of numbers or in kind of greater amount in cattle after the first 2 weeks post entering a feed yard. But whether or not those are all Mycoplasma bovis is another matter. So anyway, in that study, I think we found something like a 2% prevalence at arrival in the cattle we sampled, and we had some groups that we didn't find it at all, which really surprised me. So anyway, I don't think Mycoplasma bovis should be kind of thought of as a commensal in the same way that Mannheimia hemolytica, Pasteurella multasa, and Histophilus somni are. And Histophilus somni is another interesting one because you don't find it all the time, and it's a little harder to grow. But uh, people feel like I think they're finding it more, but it isn't always there like you can find Mannheimia hemolytica, I think. And so I've been very curious about what is it that makes a group of cattle have a problem with Histophilus somni. I think that's a lot. There's a lot we don't know about that.
Now, in some of our metagenomic investigations, we are finding mycoplasma in much, much greater relative abundance compared to Mannheimia or Pastoraliceae as a family overall, right? Now, your point is well taken that a lot of that work was done with 16S at this point, and so we really only can go to genus on that, and we need to get deeper on that. But and a study that John, Amelia, and we worked on, right? We did find that those mycoplasma levels were associated with disease during the feeding period, right? Yeah. Yeah.
But you would find that in both sick and healthy cattle.
We found it in a large population of mycoplasma in both, but more in the animals that got sick and died. Yeah.
I think it's a bit of an enigma, right? Is we've got these commensal or pathobiont organisms, and just another thing that makes this disease so interesting but complicated. I I remember hearing the question, is Mannheimia hemolytica, for instance, contagious? And I don't know if we really still know the answer to that question. It's transmissible, right? The calf probably picks it up early in life from the udder or from the environment. But in a BRD outbreak, is Mannheimia or any of the other pathobionts contagious to where an affected animal is, spreading
Yeah.
through droplets or aerosol or whatever, a very virulent strain of Mannheimia, or is it all coming from this pathobiont concept? The stress in with or without a viral infection is setting that up. I think
Yeah.
that's definitely a question that needs to be answered.
Well, and I
And
think just to say quickly, there are some papers that came out recently suggesting that certain genotypes of Mannheimia become dominant in a group of cattle after arrival, supporting the idea that it's moving from animal to animal. But I think what we all want to know is, and if that animal got disease, is that why? Is that Mannheimia that moved from another animal to that animal the reason it got sick? Or what proportion of the reason is that Mannheimia? So that's the other part of the question, right?
yeah, and we limited work again, just about to be published, that there is this clustering of those strains that I was talking about, right? So furthering what you were saying, Amelia. Yes. Yeah. Yeah.
Well, this is a question I asked you in your visit to Saskatoon. Your definition of strain, I think, needs to be are these actually just genotypic differences, or are there actually behavioral?
Yes. What is a strain? And so that, yes, genomic variant. They're a genomic variant. Yeah. What I'm calling a different is not. And there are other. If you were to do GWAS based studies, then they might look at SNP profile differences, right? And then you might look at whether or not they have gene differences for like going back to the virulence factor, whether or not there's differences there. But what I'm referring to is looking at the genome and saying that there are genomic variants. Yeah.
But so, I mean, you could do this at Vero, but just you can grow these up in pure culture, right? And why not just blow some into two groups of calves and see if there's any difference?
You're the experimental challenge guy. You and Amelia are the experiment challenge. We go out and look in real populations. So anyway, little teasing going on. Okay. So John, so the next question is for you. Speaking of this, right? So if we were to select for more resistant animals, would the viruses and bacteria just escape from what, our protection that we provided for a more resistant, genetically resistant animal? So since the viruses evolve faster than the host.
Um, well, we're ending with a really nebulous and philosophical question without any brown juice, and so I don't know how I'm going to deal
I
with
didn't
this.
ask a question. I didn't ask the question. It's on the list. Somebody asked the question online.
Again, we're all dancing around this. It's the model that's broken. I mean, cattle are very resilient animals. First of all, when we're breeding for resilience, which chromosome is the resilience gene on? I'm not sure. I know. But and then if you breed for that, then what happens to product? The whole linkage disequilibrium thing, which we talked about with the circovirus. And so, I I don't think we know, but it's all dancing around testable hypotheses without dealing with the real problem, which is the management plan, the current management plan. And so, unless and until we actually address that, I don't think we're going to actually make any progress. Especially with regard to this genetics and evolution thing on both ends, it's a huge job security kind of thing for veterinary microbiologists. But I don't know that it's going to provide any practical answers in real time. That's my short answer.
Okay. Anybody else? You're muted.
Yep, I realize that. I just want to say that um, the idea that if we select for resistant animals, won't the viruses just sort of evolve faster and then still cause disease? I do think that, whenever there's a big warm body, it's always a niche for a microbe, right? So, but I think we can't forget that even in our broken system, we have some cattle that don't get sick. The system isn't good, but it has led to a wide availability of cheap protein, right? And that's kind of what drove the whole 20th century was find ways to generate a lot of protein cheaply. If we want something different, that is animals that stay healthier, but we still want to have cheap protein, I think there are opportunities by looking at the cattle that succeed in the system. And that's what we really haven't done much of. There was work in the 70s and 80s, really exciting work that showed that co-infection or BHV infection followed by Pasteurella hemolytica led to fibrinous pneumonia. And if people made vaccines, then that didn't happen. Super exciting. We did that for 30 plus years, and it ended up not really fixing our problem. And so I think we had to try going after the microbes first. I think there are individuals that still get infected with these viruses, and they still have these pathobiont bacteria. In the face of exposure to these viruses that we all know and having these pathobiont bacteria, they don't get sick. Why is that? I mean, I would love to see the next 50 years be focused on that.
Good thoughts. Last question. How do you know which or how many cattle are, it says chronically, but I'll also add, or severely stressed at arrival? We know groups, individual cattle vary tremendously, but how would you know if they were severely stressed or chronically stressed? And I'm going to leave that open.
Open to the
Yeah,
panel.
whoever wants to answer that.
Go for it, Dr. Richeson.
How would you know if they were chronically stressed? Well, look at them. I mean, visual assessment can tell you a lot. Knowing how they were marketed and what process they went through to get to your operation can tell you a lot. Um, but just watching the animals' behavior, I know we all have limited time, but that can tell you a lot. As far as, yes, you can take blood samples and look at acute phase proteins, or cortisol is not very reliable because it jumps up and down and it moves as you handle animals. But there are things you can measure, but just I think we say this all the time, but just good old fashioned husbandry can be great, and just understanding cattle behavior and what what a healthy animal should look like and a non-stressed animal. Um, Another thing, like shrink, it can be a big telling sign, the degree of shrink, if you know your purchase weight, and then if you're able to get a weight on a truck scale at arrival at your operation. Certainly, if there is like extreme amounts of shrink, so like over 6% or no shrink can be a problem too, meaning that the cattle were probably sold as shrunk, or you have a lot of cattle that may have been like trader type of cattle. So very like no shrink at all, or lots of shrink in excess of 6 or 7% can tell you a lot about chronic stress.
So, I mean, I think the sense, or sorry, I think the technological, I really believe that we're on the verge of having commonly have a lot of data about the cattle, right? I mean, we know that there are groups that are looking at pedometers, right? How many steps they take, or they're looking at movement, how much time they move, movement patterns. Do they move back and forth from the bunk or not move back and forth, or those kinds of things. In addition to temperature and all that kind of stuff, we're probably, I believe, right on the verge of having a tremendous amount of data. And I don't know that any one of those things is necessarily going to tell us. I think some of the advanced statistical models, the algorithms that are around combinations of variables, and that probably varies depending on the types of cattle that you get in. You know, if you have imported cattle from Mexico, or do you have cattle that are coming off a backgrounding operation or whatever, it probably varies quite a bit. And I think we're probably right on the verge of having um, some data driven answers around groups, if not individuals, on that. I don't. It is my belief.
Let's hope.
Yeah.
And that we
Yeah,
can figure out how to use the data, right? The thing I worry about is that we're entering an
Well,
era of lots of data, but not necessarily actionable answers.
right. So how does that change? How does that change our management? How does that change the health outcomes in the cattle? Yep.
And
Yep.
we
it'll be
probably can't forget just the choices people make for financial reasons, right? Not necessarily at maximizing health, because they can make more money if the cattle are more likely to get sick. Um, and it's just, I think, naive not to mention that as a component of the problem system. Sometimes I worry that if we made cattle that resisted disease better, people would just treat them worse. And that's kind of an
Well, on that happy note, Dr. Willems, no, it's a complex question, complex thought process. Um, any other thoughts from the panel as we close up? I want to thank Amelia, John, and John for their time, and willingness to be able to share this around this important question. So, thank you all very much, and we hope to see you at a future event sponsored by Vero, and a panel discussion about important topics related to the cattle industry. Bye for now.